Professor. My name is Isabel Sadeghi, and here we have the Gut Microbiome Project with my partners Alex, Roya, and Dikshka. So what is a gut microbiome? A gut microbiome is specifically located within your gut, and a microbiome in general is a community of microorganisms living together in a particular environment. So I also want to explain the relationship between the gut microbiome and the physiological conditions, specifically two of them. So we have one that's the bowel condition, like the bowel syndrome, and the difference between microbiomes and a healthy bowel is that with one bowel syndrome there will be many aspects to contribute by the microbiome. The relationship would be correlated because if the microbiome within the gut is not good, then the bowel is going to be unhealthy as well. And the source that we found this from was ScienceDirect.com. Another important physiological condition between the gut microbiome is that obesity is tightly associated with specific diets and lifestyles that also contribute to the gut microbiome. Thus, an association between changes in the gut microbiota and the development of obesity has been proposed by other research experiments, which was found on ScienceDirect.com as well. So we had to change our question and our hypothesis in this experiment because our samples had not been run for the first round. So our original question was, does gender have an effect on the gut microbiome of dogs? And our hypothesis was that if the overall health of a female dog was very good, then it was due to their gender and their gut microbiome. So our new question, because our samples had not been run, was if age had an effect on the gut microbiome and their health. And our hypothesis for that was if the dog is young in age, then their overall health was very good due to the age of their microbiome. And a description of the animals that we compared was that the first two samples were a German Shepherd and an American Mastiff, which were close in age, and both had very good health, but both were different genders. The German Shepherd was a male and the American Mastiff was a female. The new two samples that we used were a flat-coated Retriever and a Husky, and they were both somewhat in the puppy range of health. They were both female. They were somewhat correlated they were both correlated the same in the weight, and they both had overall very good health. So a brief introduction to our protocol was that the first step, we isolated DNA from fecal samples using a commercially available kit, the Cohesion kit, which was specifically designed for the isolation of DNA from stool. Secondly, we did PCR, which would allow us to amplify a very specific region of the bacterial genomes, specifically the 16S RNA gene. And the amplified DNA was quantified and was provided with a unique identifying index for each sample. In our third step, it involved determining the nucleotide sequence of the amplified region of the Illumina kit, the next-gen sequencer. So our first step, which was purification of DNA, was that we isolated DNA from sterilized fecal samples using our Cohesion kit, and we isolated the DNA and concentrated that DNA with a sample of 10-fold using our commercially available kit, the Zymo kit. The isolation was based on homogenizing the stool in a buffer that prevents enzymes from degrading the DNA, and we use a series of rinses to wash away the impurities, and finally unbinding the DNA from the filter in a special buffer that weakened the bonds between the DNA and the filter. The DNA concentration also used the same idea for isolation, and the DNA was purified from the final steps of the Cohesion kit, and again bound to a filter in a column of the Zymo kit, which was rinsed of impurities and then eluted with a special buffer. And then that included that it would be 10 times more concentrated. Our second step was the concentration of DNA, and after our step with the Cohesion kit, We, like I said, rinsed off the impurities and then eluted it in a special buffer, but at 1 out of 10, the volume of the initial purification steps were making the DNA 10 times more concentration than initially. And our third step was the quantification in DNA in samples. In our second week of this experiment, we quantified and so in week 2, we first quantified and isolated the DNA and then estimated its purity. Once we knew the concentration of the DNA, we amplified the regions of the 16S ribosomal RNA using PCR, and then our expected size after amplicon PCR was 550 base pairs. For amplicon PCR, we amplified the 16S rRNA, and at the V3 and V4 sections, once the primers were bound to the temperature, it was raised to 72 degrees Celsius so that the DNA polymerase could bind and start synthesizing the DNA. PCR was conducted in a thermocycler, which was programmed to change the reaction temperature quickly. Starting with only 1 copy of the target molecule, a PCR reaction with 30 cycles could have produced many copies of the DNA, and then eventually, though, the reaction would run out of primers and nucleotides and the enzymes itself would stop to function. We would use a commercially available kit from the Promega Corporation, which combines all the reagents necessary for PCR except the template. For gel electrophoresis, it relied on the ability of charged particles to migrate in an electrical field, and we use it to separate DNA, RNA. We use it to separate DNA, RNA, and proteins of different sizes. For PCR cleanup, we use the Agincourt kit. And we follow the manufacturer's instructions as described in the lab manual. The 2 methods that we used for PCR cleanup was that we had to remove the components of the amplification PCR buffer as well as the unused forward and reverse primers, and this was critical in the success of the next indexing round of PCR, which was especially important for the removal of the unused old primers. If they were to remain in the solution, then the amplicon would have interfered with the indexing PCR by binding to the indexing PCR primers. So for indexing PCR, our PCR reaction would only run for 8 cycles. For 95 degrees Celsius, it was for 3 minutes, and 8 cycles of 95 degrees Celsius for 30 seconds, 55 degrees Celsius for 30 seconds, and 72 degrees Celsius for 30 seconds. Then we did it for 72 degrees Celsius for 5 minutes and then held it at 4 degrees Celsius. For indexing PCR, we wanted to tell which sequences came from scat samples, and to do that, we had to add a unique set of short sequence tags to the end. So, in order to explain why we did index PCR was that we needed it to determine the sequences that came from the scat samples, and in order to do that, we had to add a unique set of short sequence tags to the ends of the amplicons from each sample via the indexing PCR reaction. And when we sequence the DNA, we will know which sequence came from which samples via those tags. So for PCR cleanup, we needed to use the PCR cleanup in order to remove anything that left from the indexing PCR. We also did quantification of DNA from indexing the PCR products in order and use a fluorimeter to quantify the DNA. We used the fluorimeter rather than a spectrophotometer because a fluorometer is more accurate in its results. And for DNA sequencing, the type of instrument that we used was an aluminum next generation DNA analyzer to do DNA sequencing. The indexing PCR products were denatured, and each single stranded DNA molecule was amplified. And the single-stranded DNA molecules are attached to the surface to a small rectangular chip. The nucleotide sequence of each single-stranded molecule is then determined by the sequencing by synthesis. The instrument could tell which type of nucleotide was added, A, C, G, or T, and how many were added by the wavelength of the light produced and the intensity of the light. The instrument reported the sequence of each of the original index PCR products bound to the chip. So for initial DNA and UV spectrophotometry, our sample was 365 and 373. The concentration for our sample of 365 was 2.4, and the ratio for 260 to 280 was 2.03, and the concentration for the 373 sample was 7.1, and the ratio for 260 over 280 was 1.0/2. For our gel electrophoresis results, the sample 365 had some weak and small bands as well as negative, but, for sample 365 and 373, there was some weak and small bands as well as a negative, but positive had some of the stronger bands. And quantification of cleaning up. In our sample of 373 and 365, there wasn't enough DNA within the range, so we had a positive control, and in the positive control, we found that the concentration of 15.0 and the amount of the buffer was 90.24. We also found that there was DNA in our negative control, which wasn't supposed to contain any DNA, so in negative, we found that our concentration was of, in our negative control, we found that our concentration was 0.535, and the amount of the buffer was 77.